Okay, thank you very much. Thanks to the chairman, thanks to the organizer, thanks to Tobias in particular, who contacted me, and he kindly gave me the opportunity to talk about this topic that has recently appeared in our laboratory due to a project that we are now running and it concern liposomes and in particular extracellular vesicle. So the audience does not require to explain what does it mean mechanical measurements, but just to give you a hint, I will focus mainly on this kind of measurements where we indent a sample and objects in order to map their compliance, its compliance, or in general to obtain its mechanical properties. What is a force curve? This is also something that I guess all of you know very well. But in the anatomy of a force distance plot, what you see is that you have different portions, and the portion where we are more interested is the part where the contact mechanical details between the tip and samples is taking place. Because from this part, with different models and different, let's say from different point of view, we can extract what we need to characterize our samples. Our samples are, in general, natural extracellular vesicles. Natural extracellular vesicles now are very trendy topic of research. Just generally, probably most of you knows well them, but for sure you know what are lipid vesicles and liposomes in general. Well, extracellular vesicles are lipid vesicles produced by the cells. Well, they are produced in different districts. They can bleb from the outer surface, or they can be produced in other districts. But what is important is that they are released in the intercellular space. Their size range over different orders of magnitude, and you can call them nanovesicles, exosomes, up to microvesicles. They fly, they don't fly, they travel, and they reach other cells. They reach other cells where they are supposed to arrive, and these cells, they internalize them and they convert the message that is required. Well, they are, let's say, a universal intercellular communication tool, and from our point of view we are interested, and is our role in the project I was mentioning shortly ago, about the colloidal and mechanical characteristics, because we guess that this could play a role. And just another point, as I told you, is a message, is a communication way between cells, and to communicate you have to reach the other cell. So you have a decoration. The lipid membrane is decorated by membrane-associated protein, and these are what ensure you, what guarantee that the cell will reach just the cell that they are produced for. And then there is a cargo inside, and the cargo can be made of proteins or nucleic acids, mRNA. There is a wide bunch of objects that can be found inside, and these are the message that is supposed to be delivered. AFM is a surface technique. So, and here we are speaking about an object that has been that is living in solution. So what does it happen if we put the proteins on the surface, the extracellular vesicle or liposomes on the surface? Let's say in the first part of the talk, I will focus on liposomes because it's the way that we use to set our protocol of measurements. Forgive me for the overshooting color of this slide, but in the institute, in our institute where I tried the presentation, the projector is downgrading all the images, so the risk was to have a almost brown uniform image. So forgive me for this red and yellow extreme. But let's say, once we put vesicles or liposomes on a surface, we might have different configurations. They can stand on the top of the surface, keeping their identity. They can form or they can open up with different mechanisms. I will not go in details, but this will form a certain kind of layering. Let's say this is the typical bilayer, but you can have more than one layer on the top of the surface. But the most typical is a bilayer forming. If we put vesicles, for instance, these are vesicles coming from the prostate cancer cells. And if you put on the surface, you can have different distribution of course, of objects on the surface. But if you go in detail, this is just to exemplify what we are measuring. Well, in this case, you have a patch here that is due to the opening of a vesicle. And then on the top of it, there is a vesicle that is going to empty, is going to flatten it. So it's been damaged and is expelling the liquid. So it's like a cooking egg, let's say, forming this typical shape. If we go to make a force curve on this part here, what will we see? Well, we will see something like this. So at approximately 15 nanometers distance from the contact with the surface, we will have an increasing of the force due to the encountering three bilayers. Then if we keep pressing, we keep pushing, we will eventually punch through one bilayer, and then we will reach this level, and then we will punch through the second layer, and then the third one. So from this force curve, what we will obtain? Well, we will obtain mechanical measurements. The punch through the layers will give us hints about the fluidity and the kind of bonds which is keeping together the membrane, and I jumped. Yes. It's the other way around. Sorry. And also the distance, which is related to the topography. Well, of course, the topography you can measure even by topography itself. But this sometimes is more accurate, especially when you are dealing with very soft samples. AFM, we wanted to use to get, let's say, high throughput mechanical screening of extracellular vesicles. As usual, to be honest, you have to put your technique on the bench and then discuss which are the pros and which are the cons. So which are the advantages and disadvantages? Well, let's start from the cons. Well, the typical cons of AFM that now are changing with new models, but let's say the characteristic one is the speed. When you work with very, very soft, you might have issues of resolution. And first of all, you need a surface. The pros is that you don't need staining, you operate always in liquids, you access mechanical behavior, and let's say that this requirement of a surface might become an opportunity. Because what could be thought to be an artifact can be used to study the interaction between the vesicle and the surface. And because the vesicle is adhering to the surface via different interactions, such as electrostatic or affinity. And the objective, as I told you, is a nanomechanical characterization, and the experiments I will show you are at the first part, especially concerning these synthetic liposomes that we use to validate the protocol. Why? Because we need three important steps to have an accurate vesicle characterization. If we want to measure vesicles, so keeping their identity, so you need to optimize the deposition procedure, then you have to optimize the imaging, that is probably the less relevant part, and last but not least, the AFM for spectroscopy and the analysis. I will not go through all the trials that we have done, but we ended up with, well, let's say a very good, in the struggle between silicates such as mica, silicon oxide, we ended up with polylysine coated glass slides. And I have to tell you that with the concerning the data that I'm showing you, this is still the best solution that you can find. But we have some issues nowadays concerning so it's actually a delicate protocol because the glass and the preparation of polylysine might affect the results that you can have. So you have really to find out an accurate way to be always on a reproducible under reproducible conditions. And if we, sorry, okay. And in which way we do these experiments? Where we make images, as I showed you before, then we identify an extracellular a vesicle, a liposome. Let's talk about liposomes now. And once you identify, you do the force curves on the top of the vesicle. This is due, we do more force curves in order to compensate. I don't want to be nasty towards a competitor, but you know, sometimes you don't find a good positioning, and so in order to find really, once you make force curve, the vesicle, and in order to make force curve at different height, at different contact points. How does a force curve look like once you press on the top of a vesicle? This is the typical force versus separation curve. No contact, no interaction, then you have this linear part. This linear part is once you meet this sphere and you start to press. The first part is behaving linearly, and then there is this part. I will not talk about this part in this talk because I would run out of time, but this contains information. But this part allows us, in a very easy way, to measure by linear fitting the stiffness of the vesicle. So there are two springs, you press and you measure the stiffness. So let's say, just to have an idea, because we have to characterize the model that actually we got from the literature that has been produced, what could we change? Well, what we changed is, in the sample, is what could keep the membrane stable. So we kept the same head groups, hydrophilic head groups, and we used the different aliphatic chains. So the length of the chain is changing the stiffness, let's say the mechanical property of the membrane, and the presence of different bonds will make more or less disordered the membrane, as you can see here, saturated or unsaturated. This is also changing the mechanical properties. Some stiffness results, just to show you, looks like this. You see, this part is rather difficult to interpret, but there are in literature present some models which are making us optimistic to understand what is happening here. But let's say the linear part allows us to measure this stiffness that if we put on a scale, these are the different phosphatidylcholine that you can see here. You can see that the liposomes and extracellular vesicle can be put in a certain range that are rather understandable if you look at the different length of the aliphatic chain and the different presence of saturated or unsaturated bond. And extracellular vesicles, these are extracellular vesicles coming from the project I was mentioning you, from the group of Professor Nisum. I do not show you the objects from where they come from, because yesterday I was, let's say, harassed by a colleague of us not to show, because they are parasites from the pork gut. It's something actually disgusting, but it's important because it's something that is affecting almost the 99.2% of meat that we are eating once we eat pork meat. And this is not at all affected by the classical sterilization protocols, because they invade they attack the environment with vesicles that are giving high immunostress to the animal and eventually to people who are eating. I'm not vegetarian, so I'm not making any propaganda. I'm just saying you why we choose this disgusting object. But these vesicles are positioned, let's say, right in the middle. Is it interesting, this point? Well, somehow at the first point is important, because the stiffness is something that is affecting the deformation of the vesicle. But what is important is that the stiffness actually is made up of two contributions, so is containing two parts. Well, the first part is the bending modulus, which is characteristic of the membrane, and the second part is the internal pressure. I will go fast because I'm running out of time. And well, I made this comparison to demonstrate it. So you can have a ball which is soft skin and highly inflated, and a leather thick ball. Well, let's be fast, because you need to measure the radius of the vesicle, and from the radius of the vesicle is important. And then, by making force curve, you keep measuring the stiffness, but you also can eventually pick up a little tube of membrane, and this so-called tethering force is an independent measure of the pressure. And by a model that has been nicely developed by Gijs Wout in Netherlands, you can obtain the two parameters, and in particular the bending modulus. So the bending modulus, as you can see here, is not related to the is not as behaving as the stiffness. And this is important because the two parameters are important. In the vesicle, what is important is the presence of channels which are regulating the pressure, the internal pressure inside the vesicle. But I want to underline one thing before finishing, that we are missing extracellular vesicles. Why? Well, because this nice plateau here, which is a constant force, equilibrium, elongation of a membrane, is almost impossible to be measured on natural vesicles. Why? Because they are proteins, they are proteoglycans. It's decorated, it's not like me, it's haired, the vesicle. So once you pull it, you will have the so-called force versus elongation stretching behavior that you can see in a nice poster outside. But these that are nice from some point of view, in this case, will prevent you from measuring accurately the tether force. So this method is not working well. So we are developing another method that will not be in time to show because I ran out of time. So I will skip this slide, and I will just thank the European projects that we are coordinating about extracellular vesicles, and all the co-workers in our group. And in particular, I want to underline the role of Marco Brucale and Andrea, who are working really cheek to cheek with me to make this force distance curve. But all the consortium and all the group working on colloidal properties of vesicles, I have to thank, because they allowed us to have these highly characterized objects to measure. And thank you very much for your attention.